This is the second video of mitosis. In first video of mitosis, we have discussed what is cell division, how does it occurs, what is karyokinesis, what is cytokinesis, parent cell divide into two daughter cells, and chromosome number remain fixed for a species. Now this is the second video of mitosis in which we will study whole detail of mitosis, various stages of mitosis and significance of mitosis. So mitosis is a type of cell division where one parent cell will divide, this is parent cell and this will divide into two daughter cells. And if we start from a parent cell having chromosome number is equal to 4, 2n is equal to 4, and daughter cells will also have chromosome number 4. So this chromosome number will remain same. One nucleus will divide to form two nuclei, and this phenomena or this process is called as karyokinesis. So division of nucleus is called as karyokinesis. Now this division of nucleus takes place in various phases. First phase is prophase, second metaphase, third anaphase, and fourth is telophase. Now first we'll discuss first phase of karyokinesis, prophase. What changes occur during the prophase? So in this diagram I have shown the plant cell. Now this is the cell wall and then this is the nucleus. These are chromatin threads. What changes will occur? This we'll discuss. Now chromatin thread which is present in the nucleus here, this chromatin threads. Now these chromatin threads, they start losing water and they become short and thick. Now chromosome fiber, they condense and they will attain a definite shape of the chromosome like this. Now this nuclear membrane present around the nucleus like this will start degenerating completely. Now nuclear membrane break up into vesicles. Ultimately, formation of spindle apparatus start appearing. Now what is spindle apparatus? This we'll discuss in the next slide. So we can see here nuclear membrane is degenerating and chromatin threads, they have become short and thick. They are assuming the distinct shape of the chromosomes, and this is the diagram and this is the actual prophase seen in the permanent slide. We can see here chromosome, they are arranged loosely and they have become short and thick, right? So this is the prophase. Now we can see how they have attained the distinct shape of the chromosome. Now this central part is called as centromere and this part is called as chromatid. This is also another chromatid which is called as sister chromatid, and here DNA will be present on the chromosomes which is binded with the histone protein. Now around this spindle apparatus or spindle fibers, they start making its appearance. Now spindle fibers, they are made up of microtubules. Now we'll discuss in the next slide how spindle apparatus is produced and what is its structure. So a nuclear membrane degenerates, chromosome becomes short and thick, they assume a distinct shape and spindle apparatus start appearing. Now what are the characteristic features of spindle apparatus? It is colorless, so it is called as achromatic. This is bright and highly refractive, so birefringent. We can observe under polarizing microscope. In animal cells, here ester is produced at this end and ester is also produced at this end. So this is amphiester. In plant cell, this ester is not there. This is also not there. So this is anestral. Now this central part of the spindle apparatus is called as equator. And if it is equal on both the sides, then this is called as symmetrical, right? We can divide it into two equal halves. This is symmetrical. And if this is not symmetrical, supposing this is the spindle apparatus and one part is long, one part is small, equal part are not there. So this is called as asymmetrical. Now, if we see its constitution, spindle apparatus is made up of microtubules, and further microtubules, they may be made up of protein called as tubulin. Major part is made up of tubulin, right? So 4 to 12 microtubules per spindle fibers are present. Now we can see here, this is the spindle apparatus which is seen here, and these are the chromosome attached to the spindle apparatus. Now how many fibers are present in the spindle apparatus? This we'll discuss in the next slide, right? So here we can see this is the spindle apparatus which has almost 5 type of fibers, right? One which this is one pole of the spindle apparatus, this is another pole, right? And one which remain continuous like this part is continuous. So this is continuous fiber of the spindle apparatus. Now we can see here I've shown with different colors. This is yellow color fibers which are not continuous from one end to the another end, right? So they are discontinuous fibers. Then fibers to which chromosomes they are attached, like this white color fiber, this is called as chromosomal fiber. So this chromosome, its centromere and chromatids, they are attached to this chromosomal fiber. So it is called as chromosomal fiber. Now these two chromosomes, they are joined by fibers called as interzonal fibers. And in case of animals, fifth kind of fiber are estrol rays, right? And these are centrioles which are being produced from the centrosome. So this is the fifth type of fiber, estrol rays. So 5 type of fibers are there: continuous, discontinuous, chromosomal, interzonal, and estrol rays. So this is all about spindle apparatus. Now in prophase we discussed there is a starting of formation of spindle apparatus, and in metaphase this formation will be completed. Now chromosome nuclear membrane has completely disappeared, and chromosome are shortest and thickest in this phase of metaphase. This is the second phase of karyokinesis. And chromosome, they come to lie at the equator of the spindle. Now this is the equator which is center of the spindle apparatus, and on both the sides this may be equal, then it is called as symmetrical. So chromosome, they come to lie at the equator of the spindle apparatus. This is also called as metaphasic fluid. And we can see here this part is centromere and these are the chromatids. So it get attached to the chromosomal fiber with the help of its centromere, and this form a metaphasic plate or equatorial plate because it is being produced at the equator. Each chromosome is connected to both the spindle poles by separate fibers called as chromosomal fiber or tractile fibers. Now when this metaphasic plate has been produced, then it will start preparing for the next phase of the karyokinesis, that is anaphase. So a protein complex which is called as anaphase promoting complex, this will become active in this phase of the metaphase. So this is the diagram, and we can see here this is from the permanent slide. Chromosome, they have become short and thick, and they are arranged here at the equator of the spindle apparatus here, right? So this is metaphase stage of the mitotic cell division, right? Now chromosome in metaphase, they are arranged in the equator of the spindle apparatus forming a metaphasic plate. Now this will enter into the third phase of cell division which is called as anaphase. Now what happens, what changes occur during the anaphase? Like we have seen here, this is the centromere which is attached to the chromosomal fiber. Now this centromere start dividing into two in the anaphase phase. So we can see here the centromere has divided into two, and this is also adhered with the help of will be attached with the cohesion protein which is adhering the two centromeres, and these are two chromatids like this and like this, right? So centromere divides into two. Each chromatid will comes to have its own centromere, and these cohesive protein, right, which are attaching the centromere, now this will be degraded and these two chromatids will get separated. When this part is degraded here with the help of anaphase promoting complex, which will bring degradation of the cohesive protein, and chromatids will get separated. So they will start moving toward the chromosomal fibers toward the upper pole, because this is also made up of tracin and actin, which help in contraction of the chromosomal fibers. So we can see here in this figure, two groups of chromosomes are present at two poles. So this is called as anaphase phase. So this in early anaphase they start separating, right? And then they start moving along the chromosomal fiber toward the two poles, right? This will move toward this pole, this pole, this pole, and this will move toward this pole, right? So they start moving toward the pole of the spindle along the path of the chromosomal fiber, right? So this is middle anaphase. Now in the late anaphase, they will reach toward the pole, right? Now they have reached toward the pole because of the contraction and contraction of the spindle poles toward the polar part, this pole part, this pole part. So these chromatids, they will move toward the pole of the spindle apparatus. They will move in the opposite directions. So they are reaching toward the poles, right? So this is called as late anaphase. Now the cell will enter into fourth stage of karyokinesis. It has almost reached at the pole of the spindle apparatus, top of the spindle apparatus, right? Now nuclear membrane may start nuclear membrane will start appearing around the these are chromatids, right? So nuclear membrane will start appearing around these chromosomes, right? When nuclear membrane start appearing, this is from the cell organelle called as endoplasmic reticulum, right? So when this membrane has started forming, right, now spindle fibers may start disappearing at this end. So this is the early telophase where they have reached at the top, and then nuclear membrane have start appearing. Now we can see here this part, this is the telophase, right? Now this is the equator of the spindle apparatus, and these are fibers we can see here. Now these fibers are visible here, and these chromosomes, they have reached toward the two poles. And this is the anaphase, and this is the telophase. We can again see here loosely arranged chromosome prophase. Now when it has started forming in the center, this is metaphase, reached toward the two poles, this is anaphase, and now this has reached toward the poles, and ultimately this is early telophase. And when this will end, this will bring, and this will divide into two nuclei, then this is called as late telophase, right? So here we can see nuclear membrane has been completed, right? This will be completely produced, and nucleolus which was degenerated into the prophase stage, now this nucleolus will be produced again. So this formation of the nucleolus is also called as nucleologenesis, right? Like earlier, when nucleolus degenerated, its remnant particles will be there in the cell wall materials. Now those in the cell cytoplasm materials, they will again become accumulated and form the nucleolus. So this is called as nucleologenesis. Nuclear envelope formation has been completed. So one nucleus, we can see one nucleus has been divided into two nuclei, right? So this is called as karyokinesis, and there are various phases like prophase, metaphase, anaphase, and telophase. So ultimately we have got from one nucleus two nuclei. So karyokinesis has been completed. Later on, cytoplasm may divide. Now this may divide into like this may divide into two cells, and cytoplasm divides. This is called as cytokinesis. This we'll discuss in the next video. So one cell has divided into, this is one cell, this has divided into two cells, right? So this will be one from one parent cell, two daughter cells have been produced, and chromosome number remain fixed for a species. So these are various phases. Again, we can see this is prophase, where chromosome, they are loosely arranged. Here they are arranged at the equator of the spindle apparatus here also. And then we can see here, this is the prophase. This is we can say early telophase, and from one cell two cells have been produced. This is the late telophase. So this is all about mitotic cell division. If you have any questions, you can ask me in the comment box. Now next is, what is the significance of mitosis? What does it signify? Mitosis plays a very important role in the growth and development of an organism. Supposing we have sown a seed, right? And that seed may develop into germinate, and this will develop into plant. Ultimately, we may get a short plant, right? And this is the root system. Now, this will help in growth of the plant as well as animals, right? So, growth and development is occurring with the help of mitotic cell division. Now, when one cell divide into two cells, right? Now, this cell will further mature up to attain a proper size. So, cell size is maintained by mitotic cell division. Now, if we see this is one cell and it is having nucleus, right? And this part is cytoplasm. So, this is called as a Caryoplasmic ratio or nucleocytoplasmic ratio. I mean, what is the amount of nucleus? What is the cytoplasm? When one cell divide into two cells, now they are short. Later on, they will become mature and they will attain proper karyoplasmic or nucleocytoplasmic ratio. Now, if parent cell has chromosome number 4, daughter cell will also have chromosome number 4. If parent cell has chromosome number 16, daughter cell will also have chromosome number 16. So, chromosome number remain fixed for a species and there is maintenance of the chromosome number throughout the generations. Now, mitosis also helps in maintaining genetic similarity and regeneration. Like if we are, and this also helps in asexual reproduction, like in fungi and lower organism. Reproduction is with the help of asexual method, by conidia, by sporangia formation. Their mitotic cell division take place. And like if we could talk about vegetative propagation in the plant, like if we sow potato, we will get potato crop. If we sow sugarcane stem, we will get sugarcane crop. If we take a leaf of sansevieria or bryophyllum, we can raise the plant of sansevieria from sansevieria leaf. We can raise the leaf of from leaf of bryophyllum plant. So this will be with the help of mitotic cell division or by mitosis. So this helps in maintaining the genetic similarity and regeneration. Supposing we have a this also helps in healing, right? Supposing we have a lizard whose tail has been broken. Now this can get regenerated with the help of mitotic cell division. If there is any wound or cut in plants or in animals or human beings, that can be again healed with the help of mitotic cell division. Now, if we talk about all the living beings on the earth, whether they are vertebrates, invertebrates, mammals, insects, plants, various types of plant, basic pattern of mitosis is universal. This will remain same in all the living organism. So this helps in maintaining the basic similarity. So this is all about significance of mitosis. Thank you for watching my video. If you like my video, please like, share and subscribe.